successful future. So the question is, can animals do that? And again, it looks like at least some species of animals can. Again, the scrub jay is the leader in this enterprise. And the experiment that was done was to prepare two alternative dormitories for these scrub jays. In dormitory A, in one part of the lab, they were kept overnight, and in the morning, they were just left there. They weren't given breakfast. In the other dormitory, which was had different internal furnishings and so on, so they could distinguish the two, they always got breakfast. So now scrub jays were given a little light supper of a bunch of peanuts. If they were in the room where they never got breakfast, they hid those peanuts. If they were in the room where they always got breakfast, they ate the peanuts. They seem to have this ability to project themselves forward to the next morning, and on the basis of where they were, decide what to do. Okay, so that's the a sort of overview of how we think about the structure of the various sorts of memory that the brain can encode. How does it do it? So now we need to talk a bit about the machinery of the brain, the actual cells that are doing all this storing and retrieving. And this, of course, is largely the neurons, the computing elements of the brain. And neurons come in a wonderfully wide variety of shapes and sizes. Here are two important classes of neuron in the next slide. So here we have a pyramidal cell. It's called pyramidal because although you can't really see it in this slide, and this is one of Cahal's drawings, the cell body, which is here, is elongated like a pyramid. And these long processes here are the dendrites, the receiving parts of the cell, to which other cells project. And also down here, this is mostly dendrites. There's a little axon, which is the fiber which transmits the nerve impulse to the target cells of these pyramidal cells. This beautiful thing is a Purkinje cell of the cerebellum, part of the brain that allows us to move and balance and so on. And that's a drawing of one, and here's an actual cell which has been filled with a fluorescent dye. And you can see the electrode here, which is penetrating the cell body, and dye is diffusing throughout the cell. And you have to imagine that everywhere on these dendritic processes, there are little stubs, little synaptic spines, which are the receiving parts of the cell onto which excitatory neurons project. So here's a pyramidal cell, and if we magnify one of these dendrites, we see that it looks like this. And these little protrusions are what Cahal calls synaptic spines. And each one is a postsynaptic element. That's to say that we don't see, but there's an axon coming along here, forming a synaptic bouton, or a presynaptic element as it's called, which excites the target cell by releasing transmitter onto this little spine. And embedded in the membrane of this spine are specialized proteins called receptors. And when the transmitter binds to the receptor, it excites the target cell. So I'm going to switch to a diagram now to explain that. So here is an axonal terminal. This is an axon. You can imagine an action potential or a nerve impulse traveling along this axon. And what happens when it gets to the nerve terminal here is that one of these little vesicles, spherical vesicles, fuses with the presynaptic membrane. And each vesicle contains a packet of transmitter Transmitter is a chemical substance which is going to bind with the receptors on the postsynaptic cell. The transmitter at excitatory synapses is called glutamate, and there are various sorts of glutamate receptor which play different roles at different times. The receptor type which transmits excitation normally is called an Nmda receptor, and it's got a little channel in it. And when glutamate binds to the Nmda receptor, the channel opens and sodium ions flow into the cell. And the consequence of that is to produce a reduction in the membrane potential across the cell membrane. And that's an excitatory event. And if there's enough excitation of that sort, a threshold will be reached and the target cell will fire a nerve impulse. There are other sorts of receptor which play an important role in our story, and their name is the Nmda receptor. But normally they don't play much of a role during normal synaptic transmission. And the reason for that is that although glutamate binds these receptors, the channel is blocked by a magnesium ion, and nothing can get through, so nothing happens. I want to talk briefly about numbers. How many cells are there in the brain? Well, there are about 100 billion neurons in the human brain. If you talk about synapses, and synapses are really the individual computing element of the brain, there are at least 1,000 times more than that. So we're talking about 100 trillion synapses. If the brain were just digital, if each synapse was either on or off, as it were, we might ask ourselves how many memories could, say, the hippocampus store? Well, the number of neurons in the hippocampus is about 10 million in humans. The number of synapses per neuron is greater than the average for the whole brain, because those pyramidal cells have thousands, if not tens of thousands, of synaptic spines. So let's say there are 10 to the 11th synapses. And let's say, under our supposition that synapses are little computing devices which are either on or off. And let's suppose that every different configuration of synaptic weight in that synaptic space represented one memory. Then the number of possible memories would be 2 to 10 to the 11th, which with a little jiggery-pokery gives this number here. How big is that number? Well, it's pretty big. The number of particles in the universe is 10 to the 88th. That's less than 10 to 100, which equals 10 to the 10 to the 2, and we're looking at 10 to the 10 to the 10. So there's plenty of space within the hippocampus for a vast number of memories. And that's with a digital synapse. But synapses are not digital. They're little analog machines. And every individual synapse is a complex machine in its own right, containing thousands of proteins. So to come back to the hippocampus and our search for the Hep synapse, where are these Hep synapses, which everybody says must exist? The next slide shows the other thing which made it seem like a good idea when I was starting this work 30 years ago, 40 years ago rather, to look at the hippocampus, was because the hippocampus is a simple structure compared to the neocortex. So what I'm going to show you now is a transverse section through the rat brain. And you can see here that there's not much evidence of organization in the neocortex, but come down to the hippocampus, which has moved up in the brain, or rather it's being pushed down in humans, it's up here in rats. You see basically there are two sorts of cells. This inverted C bunch of cells here are called granule cells, and these are the pyramidal cells of the hippocampus. Okay, so it's a simple structure. Does that matter? Well, as we'll see, it matters a lot from an experimental point of view. So just to look a bit more closely at the organization of the hippocampus. And we've got these tightly packed cell bodies. Information comes into the hippocampus from a part of the cortex called entorhinal cortex. And what comes into the hippocampus is a version of what's going on in the external world. It's a highly processed version, but the hippocampus is getting information about what's going on. That comes in first to the granule cells, and that's this arrow here, so activity is coming along here to granule cells, and then it's being propagated by action potentials or nerve impulses along this pathway, which ends here. And these cells, these target cells, are called CA3 cells, and finally the CA3 cells project to CA1 cells. And so you get this, this sequence of excitatory pathways, and information is being processed in the hippocampus in that sequence. It comes in here, and then there, and then here. And each of these is what's called a monosynaptic, just one type of synapse. So we've got 3 monosynaptic pathways. So it starts off, I'm looking down here in the lower right here, perforant path, first synapse projects to granule cells, the axons of the granule cells, second synapse project to CA3 cells, and the CA3 cells project to CA1 cells. There's something else which is highly attractive to the experimental scientist about the hippocampus, and that is the way that, the pathways that come into the hippocampus are very tightly organized, very highly laminated. So if we look at this pathway here, this perforant path which comes in to the granule cells, there are actually 2 components of the perforant path. This is a cross-section of the hippocampus stained for heavy metals in nerve terminals. It's a sort of specialist sort of stain. And what it picks out so beautifully is that in this region here, where the dendrites of the granule cells extend, there are 3 main inputs, which are characterized by the amount of zinc or lead in their terminals. That doesn't really matter to our story, but it does matter that the organization is so precise. I mean, it is really remarkable that the fibers coming from the lateral part of the entorhinal cortex will only go to the outer part of the dendrites of these granule cells, and the medial perforant path only terminates on the middle part. And the reason why that's important from an experimental point of view is that it enables